Hello and welcome back to Biology for Beginners. As always, I'm your host, Morgan. Before we jump into the topic, please remember that Chapter 12 is a little bit heavier and a little bit more technical than the other chapters in Unit 2. So if what we discuss here feels a little hard to digest at the end, that's perfectly okay, and most of what we talk about here an entry-level bio student, or at least a non-major entry-level bio student, wouldn't be expected to know most of this material here anyway. And a lot of the topics are kind of going to be glossed over, mainly because I don't want to overwhelm you, and your average entry-level class isn't going to expect you to know it anyway. So today's topic is genetically modified organisms. As always, the terms and definitions should be popping up about right now so that you can pause the video and grab the vocabulary we will be using today. Or if you want the entirety of my slideshow as a PDF, please feel free to hop on over to my Patreon. The link is in the description below, where all of my Biology for Beginners slideshows and any supplemental study aids that I have made to go along with these videos and to help you in your studies you can grab for absolutely free. However, if you do become a patron, you get the Bio for Beginners content at least a day in advance. Before we get into the new material here, recall that genes can be easily added to plasmids and inserted into bacteria, usually E. coli. And then you can rely on that bacteria to reproduce and make more copies of that gene. And then you have two possible paths. Either the protein products of that gene can be harvested and used, or the gene can be inserted into another organism. But one thing to note is that bacteria are not always going to be the best choice if you're looking for a particular protein product, and sometimes you have to use other cells. So that brings us to glycoproteins and the utilization of mammalian cells to produce those glycoproteins. So E. coli can't make glycoproteins the right way. And it's really just because the way that certain mammalian proteins and other organisms' proteins as well are structured, the single-cell organisms don't really have the correct machinery to make them. So when that happens, we're specifically going to stick to this mammalian example. So mammalian cells need to be used to make glycoproteins. So both cell cultures and entire animals can be used to do this. And if you are using an entire animal, you end up with something known as a genetically engineered animal, because you have genetically engineered it to produce a specific thing. You've messed with its genetics a bit. So one example of a genetically engineered animal that's used to produce something specific is a human protein that prevents blood clotting is inserted into goats and harvested from their milk. And this can then be used to make medications and things for patients suffering from stroke and other blood clotting diseases. And so genetically engineered animals aren't really the norm. Usually, if you're going to need a glycoprotein that needs mammalian cells to produce it, you're just going to use a cell culture. And the reason for this is that it's difficult and costly to produce, so they're only going to use that option of the entire animal when necessary. If you go back to our cloning video, it's going to have some similar complications that cloning has. So it's expensive and it's difficult, so it's only going to be done when absolutely necessary, like in the case of the protein that gets harvested from the goat's milk. So this brings us to the question of why are we going to use DNA technology and why are we going to be messing with genetics? So one of the big deals of DNA technology is treating diseases. One of the big kind of successes of disease treatment using DNA technology is the production of human insulin to treat diabetes. Prior to being able to produce this human insulin in the lab, insulin from other sources, specifically pigs and cattle, were used. However, because it wasn't human insulin, people could have allergic reactions to insulin sourced from other species. So it was a big deal when they were able to produce human insulin through these genetic manipulations. Another thing is DNA technology can be used to diagnose diseases. And the way they do that is you can identify the presence of certain genes and potentially the organisms responsible for the diseases. And then you can also use DNA technology to prevent diseases. So the big heavy hitter here, the prevention world, is using this DNA technology to produce vaccines. And especially in the time that this video is being recorded and released, we are seeing DNA technology being used to produce a very important vaccine. But we're not going to go more in depth into that because I don't want to open that can of worms when we're just talking about why we use DNA technology. The thing that most people probably recognize DNA technology from the most is DNA technology and agriculture. And this is going to be kind of the main event of this video. This is where we're going to talk about genetically modified organisms, or as you know them, GMOs. So this is where we've used DNA technology to improve the productivity of both plants and animals. However, it's mostly plants, so we're going to talk about plants. So basically, a GMO is an organism that has acquired one or more genes by artificial means. So that means that in the lab, we've inserted genes into the organism. And if the gene is specifically from another species, the particular GMO you create is called a transgenic organism. And that just means the genes that have been inserted are from another species. So if you think back to our video from last week with the glowfish, that is technically a transgenic organism because it is a fish with genes from sea jellies inserted into it to make it glow. But back to plants. So GMOs are going to most commonly be plants, as I've mentioned, and the most common modifications are going to include things like increasing production, or increasing nutrition, or increasing pest resistance. You see, there's a theme here. You make these plants a little bit bigger and better. And let's talk about the steps here. So you're going to get your target gene from whatever organism it's going to come from, and you're going to insert it into an agrobacterium. Now, that's the particular genus of bacteria that's used for the plant stuff, and that's just because this bacterium is the best choice for this world. If you want a little bit more about the actual gene manipulation process, please review last week's video. And so once that target gene is inserted into the bacterium, you take that recombinant bacterium and insert it into a plant cell. And then the DNA is going to combine with the plant chromosome, and then that cell can be cultured and you grow a new plant with a new trait, and that's your GMO. This brings us to the question that is on a lot of people's minds, and the question that you've probably seen in media, and the question that makes those fun little stickers on certain food products in the supermarket say non-GMO or no GMOs. The question is, are GMOs safe? And something you need to know before we really talk about the yes or no is that most of the staple crops like rice and corn have been genetically modified in some way, and at least if you're in the United States, you don't necessarily need to label whether you use a GMO or not in most cases. And again, if you are in the United States, you've probably eaten a GMO today. But that's just because most of our staple crops have been genetically modified in some way. So on to the question of safety. Now, let's first tackle, are they safe to eat? So in 2012, there was a study published done on pigs, and the control group of this study was fed non-GMO corn, and the experimental group was fed 39% GMO corn. Now, why use pigs? Well, the digestive system of pigs is fairly similar to that of humans, and in general, if you're doing studies like this, pigs are a good human stand-in, since it's kind of unethical to do studies like this on humans, and there is a whole question of ethics in general in the science world when you're working with the animal kingdom, specifically chordates. And chordates are things like you and me. Mammals belong to that little grouping, but we'll talk about more of that stuff when we get into evolution and all of that fun. But now we need to get back to our question here is, are GMOs safe? So the overall conclusion of this study, once it was run, once the health of the pigs was tested at various stages in their life, and once they were slaughtered and had their tissues tested for foreign DNA, was that there was no difference in the health of the pigs that were fed the control non-GMO corn and the experimental 39% GMO corn. And the tissue that they tested post-slaughter showed absolutely no foreign DNA. So they didn't pick up any of that inserted genetic material from their food. It just was food. I know that there's a lot of controversy specifically around studies like this, saying, oh, well, that hasn't been done on humans, so we don't really know for sure. But overall, the science comes back to conclude that, yeah, GMOs are for the most part safe to eat. So at least breathe a sigh of relief that your staple crops and the foods that you eat, you're not going to get sick just because they've been genetically modified. However, there are some environmental concerns that are also raised when you talk about the question of safety. And the big thing, the big question mark, the big concern is, can GMOs become loose in the environment? And this question is more maybe. And it's basically because there has been some evidence gathered of unintended gene transfer to wild plants. But this kind of needs to be looked into more, and this is more of an open-ended question. So yeah, there are some environmental concerns. But the big conclusion here is that GMOs need to be evaluated on a case-by-case basis. And you can't take any study, even if there are studies out there that say this particular GMO is unsafe. That doesn't mean all GMOs are unsafe. It just means this one was a flop. But on the other side, you can't take one study saying this is absolutely safe, so that means all GMOs are safe. Again, it needs to be case by case. And as new genetically modified organisms come out, there needs to be certain standards to ensure their safety, both for consumption and both for the environment. So are GMOs safe? Some of them, for the most part, yeah. So don't freak out when you hear genetically modified organism or see that there's a GMO in a particular food item, although you're unlikely to see that since they don't necessarily need to label it anyway. So our last little bit of this video is entirely hypothetical. And this is talking about modifying the human genome. So the big deal here, and it's controversial because, again, when you do work on anything with humans, there's ethics to consider and all that stuff, and we're not getting into the ethics, we're just getting into the hypothetical process and possibility of gene therapy. And so gene therapy, the big idea here is that you could alter genes of a diseased individual to basically treat the disease. And the hypothetical process here, using bone marrow as an example, would be that one, you would get a gene from a healthy donor and clone that gene and then insert it into a retrovirus. Remember, retroviruses have a specific enzyme, reverse transcriptase, that can take mRNA and synthesize DNA from it, completely reversing the normal flow of genetic information. And then, since you'll have that gene from a healthy donor, you'll insert that retrovirus into a bone marrow cell. And then the viral DNA and the human gene can then end up inserted into the chromosome. So the idea here is that you would then have the healthy gene. And then any engineered cells from this process can then be injected into the patient and hopefully treat their disease. Again, this is entirely hypothetical right now. This is not something that is done, but it is an area of research. There are ethical concerns around it, but we're not here to discuss the ethics. We're just here to talk about the hypothetical technology that's being talked about here, because gene therapy could be a potentially very good thing for disease treatment. But yes, there are ethical concerns and all that stuff, and when you're working with human genes or working with human anything, there are concerns. But that's where we're going to end today. If this video was a little hard to digest, I completely understand. The big deal in this video would just be knowing the process of getting genes into plants to produce a GMO. So that 1 or 2 slides is pretty much what any normal entry-level bio class would expect you to at least have some idea of. But I do hope you have a wonderful rest of your day, afternoon, evening, what have you. Please remember to like, comment, subscribe, all that fun stuff, and I will see you all in the next one. Thank you again for watching this video. Don't forget to like, comment, subscribe, and ring that bell if you would like to see more. And if you'd like to follow me on any of my other social medias, the links are down in the description below. Don't forget to check out thereptilegoth.com for all of my articles and blog posts. If you found any value in this video and you would like to help support the channel, please check out my Patreon page. That link is also in the description down below. And a special thanks goes out to my Diamond Dragon patron, Diane V. What you're doing is really helping me fund a dream here. I will see you guys all in the next one.